3, 2, 1.
Hello, welcome, and thanks for joining us here for our weekly seminar series hosted by the Living Earth Collaborative and Washington University's Ecology, Evolution, and Population Biology group. I have the great pleasure to introduce our speaker for today, Dr. Emily Warschefsky. Dr. Warschefsky is an evolutionary biologist who focuses on crop domestication, and currently we're very fortunate to have her here in St. Louis as an associate scientist with the William L. Brown Center at the Missouri Botanical Garden. Dr. Warschefsky's academic career started at Florida International University, where she graduated with a PhD in 2018. During that time, she held a prestigious doctoral fellowship through the U.S. Department of Agriculture. After finishing at FIU, Dr. Warschefsky joined the Department of Botany at the University of British Columbia as a postdoctoral research fellow, where she was until 2020. She then moved to the University of Colorado Boulder on another prestigious postdoctoral fellowship, and then finally joined us here in St. Louis, where currently Dr. Warschefsky is the Alice Brown Curator at the Missouri Botanical Garden. Dr. Warschefsky's research is focused on the evolutionary domestication of crop plants, particularly woody perennials, and the conservation and use of their crop wild relatives. Already in her career, Dr. Warschefsky has racked up an impressive array of accomplishments, including the multiple pre- and postdoctoral fellowships that I mentioned before, as well as an excellent record of publications, including in journals like the American Journal of Botany, New Phytologist, and Nature Communications. Her research accomplishments have even led to press coverage in National Geographic and Newsweek. I think many of us here in the city view Dr. Warschefsky as a scientist whose work we're all going to know and discuss in the not so distant future, and we feel grateful to have gotten her here through Mobot, and especially grateful that she agreed to give a talk in our seminar series today. So one last thing before I turn it over to Dr. Warschefsky, I'm going to remind you all in the audience to leave any questions or comments that you have in the chat section that you see next to your video, and the host will relay them to our speaker at the end of the presentation. And so with that, without any further delay, I'm going to turn it over to you, Dr. Warschefsky.
Thank you very much. I'm really excited to be here speaking with all of you at the Living Earth Collaborative and to be here in St. Louis locally. I really look forward to getting to know you and hopefully form some great collaborations. Today I'm going to tell you about my research program and which focuses on the evolution, domestication, and improvement of diverse crops. But I know I'm talking to a pretty broad audience, so I want to start out by telling you about why I work in domesticated systems. And the first reason is that because of the close relationship between humans and domesticated species, the study of crops is inherently the study of our own human history. These species provide a window into our past. When humans move around the world, we take food plants with us, and they're an important part of our culture. Even today, we still have events like harvest festivals. But beyond this human element, domesticated species have played a really important role in the advancement of evolutionary biology and genetic theory. In fact, in The Origin of Species, Darwin spends his entire first chapter focusing on variation under domestication and reasoning that this variation he observed in domesticated species was a result of human selection, and that mirrors the selection that happens in nature. He later went on to write an entire book on domesticated species. And of course, Mendel famously used domesticated species to first explore the principles of heredity and lay the groundwork for the field of genetics. Today, domesticated systems still provide us excellent tractable groups in which to study both evolution and genetics. But in addition to being these great study systems and helping us learn about our own past, domesticated species are critically important to the future of global food security. In the face of changing climatic conditions and erratic weather events, we as a society need to figure out a way to produce more food more sustainably in order to meet the demands of our growing human population. So my research falls into the category of domestication genomics. And this is an area that I consider to have three main goals. The first is to understand how evolution and domestication have shaped modern crop diversity from the past events. Second, to conserve this diversity that we still have today, and then to use that diversity to make agriculture more sustainable for the future. You might notice that there's a bit of redundancy here. I use the word diversity in each of these aims, and that is really at the heart of this field. But for many years, and even to a great extent now, a lot of our research on crop genetics, crop domestication, crop improvement have focused on a relatively small number of important crop plants like corn, wheat, maize, soy. And although these are certainly really important for our food security systems, they do not nearly encapsulate the diversity of species that humans have domesticated over the past 12,000 or so years. From fruits and vegetables to spices, nuts, oil plants, and of course, some of our favorite things like coffee, tea, and chocolate. We have domesticated a phylogenetically, geographically, and ecologically diverse domesticated flora. And my work seeks not only to improve our understanding and use of diversity in traditional crops, but also to expand our knowledge about how different types of crops, particularly woody species, are impacted during the domestication process. And so woody crops are actually perhaps more important than you might realize. Of the world's 50 most produced crops, they represent 1/5 of these species, and woody crops are 12 of the most valuable species worldwide. It's also becoming increasingly clear that these woody perennial species can and should play a critical role in the future of agriculture. From agroforestry programs to urban food forests, trees provide a versatile and sustainable component of our agricultural systems. My primary study system has been the genus Mangifera, which includes the domesticated species mango, a single species, Mangifera indica. This is a fruit with an amazing array of color and shape and size, as you can see here. Unfortunately, what you can't tell from these pictures is that these fruits also have a great diversity of flavor. If you've been lucky enough to travel to a part of the world where mangoes grow, you hopefully have had a chance to experience this yourself. So the genus Mangifera is a group of about 70 species, making it the third largest genus in the family Anacardiaceae. This is the family that also includes poison ivy, pistachio, cashew, and sumac. And these species are all from South and Southeast Asia, with a range that extends from India eastward to the Solomon Islands. But the area of highest diversity, as you can see in this heat map, is Borneo, Sumatra, and the Malay Peninsula. Mangifera species tend to be quite tall trees, getting up to 40 meters in height. They grow in wet lowland tropical forests at relatively low densities and are often irregular in flowering times, with some species flowering as infrequently as once every decade. So for this reason, they're poorly represented both in living and herbarium collections because they are difficult to locate, to collect, and to identify. As a PhD student, my research focused on understanding the evolution and domestication of mango and its wild relatives within this genus. And for this project, I visited a number of botanic gardens and institutions in Southeast Asia to make collections. I gathered around 280 individuals representing 35 taxa. Around half of those collections were individuals of cultivated mango. And I used RAD-seq sequencing to examine the evolution and domestication of this group. It's a great tool for the questions I was interested in because it can analyze data that spans from deeper scale phylogenetics to more recent population genetic questions. So I'll start out telling you a little bit about this deeper scale time, what I like to call a fruitful phylogeny. And it's not just fruitful because it includes mango, but in fact, the group that I was looking at, Mangifera and its sister genus Buia, included almost 30 edible species between the two. So the primary questions I was interested in were somewhat simple because this was the first multi-locus phylogeny of the genus Mangifera. Essentially, I wanted to know, how are these species related? And does that align with the morphological hypotheses that we've made about relationships within this group? Which species are closest relatives of Mangifera indica and might be useful in breeding programs or as rootstocks? And then because there's this large number of cultivated and edible species, how are those species related? Do they come out in a single clade or are they found in multiple groups? To answer these questions, I produced a maximum likelihood phylogeny. And I did this with varying numbers of individuals. This figure shows a subset of samples of 41 individuals representing 35 species, including an individual from all subgenera and in each section. However, I produced phylogenies that included as many as 201 individuals. And regardless of the sampling and replication within species, I consistently recovered these 5 well-supported clades. These were also found using another method of phylogenetic analysis, quartet-based methods called SVD quartets. So this first question is looking at whether the morphological hypothesis within Mangifera corresponds to what we found in the phylogeny. And the morphology showed that Mangifera could be delineated into 2 subgenera and a total of 6 sections. To tell whether the morphological and genetic classification align, I mapped these hypothesized placements onto the phylogeny. So in this, the dark gray shows the outgroups. The light gray shows individuals that were not classified previously by morphology. And then all of the remaining colors correspond to subgenera and sections. And it's pretty apparent that the morphology does not follow what we found in the phylogeny, although there are some patterns, especially when you look at the level of subgenus. We do see some differentiation between these color groups. So none of these previously described sections or subgenera are monophyletic. But if you're paying close attention, you will also notice one real oddity, which is that there's a clade of species within Mangifera that falls outside of the sister genus. So within these 2 outgroups. This was not at all expected, but it turns out that once we looked at the morphology within this group, we found that deserves a designation as a new genus of Anacardiaceae. So this is a project I'm working on describing this group with a collaborator from Singapore Botanic Garden and also Carolina Romero, who works in the William L. Brown Center with me. The really kind of interesting thing about this group is that these are not species that were unknown. In fact, some of the species that will be placed in this new genus include highly cultivated species, some of the most common backyard fruit trees in Malaysia. And it's pretty clear once we looked at the morphology, you know, based on some of the floral characteristics and the fruit. As you can see here, they almost look more like potatoes than they do mangoes. This does deserve to be classified as a new genus. So, okay, what about the mango clade itself? What species are related to mango and might be agriculturally useful? In this phylogeny, mango appears at the very top where the red arrow is pointing to, and it comes out in a clade of closely related species that include, at least in this sampling, about 8 different species. These include really interesting things like Mangifera gadibi, which is shown in the kind of bottom picture of the square. This has flattened fruits that are water dispersed, and Mangifera casturi, which has the purple skin in the picture, and the flesh of this fruit is really rich, bright orange color. So it's a group that has some potential for use in agriculture. In terms of the cultivated species of Mangifera, it turns out that in each of these 5 major lineages of Mangifera and Buia, there are cultivated species and wild species. And I was particularly excited about this result because, to me, it demonstrates that Mangifera could serve as a novel system in which to study domestication. If you think about domestication as being a continuous process, species of Mangifera that are cultivated span the breadth of this spectrum, from truly wild to sort of semi-domesticated, wild collected, backyard grown, to rather highly cultivated species like the mango. And therefore, they offer an opportunity to explore parallel domestication in action, and in a tropical tree system, no less. So I'm looking forward to continuing this work within mango. So zooming in a little bit to think just more specifically about the history of cultivated mango, I looked at the domestication of Mangifera indica, and this is work that was published in New Phytologist, if you're interested in checking that out to get more detailed information. So the mango was originally domesticated in India, and it's hypothesized that the origin, the wild origin of mango was somewhere in the foothills of the Himalayas in northeastern India, Bangladesh, Nepal, and Bhutan. We're not entirely sure if those wild populations still exist or whether they truly represent the actual wild progenitors. Domestication, we believe, happened around 4,000 years ago. And then from India, mango was first introduced into Indochina, here shown in blue, and Malaysia in green, sometime in the 4th and 5th centuries. And all of these dates are coming from historical documentation and archaeological sites. This introduction happened likely by Buddhist monks who brought mangoes with them during their journeys. It wasn't until much later, in the 9th or 10th century, or perhaps as late as the 16th century, that mango was introduced to the West, first into East Africa, and then into Western Africa. And here it was just being passed along the typical trade routes of the day. In the early 1700s, the Portuguese introduced mango into the New World, first into Brazil, and then it made its way into the Caribbean in the mid-1700s and into Mexico in the mid-1700s, shortly thereafter. Mexico is particularly interesting because it received introductions of mango both from this westward migration out of India through Africa and through the Caribbean, but then also directly from Southeast Asia through the Philippines. And this is because both Mexico and the Philippines were under Spanish rule at the time. And then finally, the first recorded introduction into Florida didn't occur until 1833. But since that time, South Florida has become an important area for mango breeding and cultivation, particularly in the earlier 1900s. And many of the world's most important mango cultivars, commercially speaking, were actually developed in South Florida. So why is this history of migration and movement around the world so important? Well, it's because typically when we think about diversity during domestication, we think about domestication bottlenecks. And that's this process where when we start out with a relatively diverse wild species, we see this primary bottleneck that occurs during the domestication process. And that results in traditional crops that have slightly less diversity. And then a secondary bottleneck occurs during modern breeding and improvement. And that oftentimes means that our modern crops have fairly low levels of diversity and are likely susceptible to disease outbreaks and are not able to tolerate harsh abiotic conditions. However, the story is slightly different in perennial crop species. And in 2014, Brianna Gross showed that in most perennial crops, they are able to maintain relatively high levels of genetic diversity during the domestication process. However, our sample size for this data it's pretty limited. And so we want to know more about this and how perennial crops respond to domestication, especially different types of perennial crops, including tropical trees like mango. And so while I can't take a good look at the primary domestication bottleneck that occurred because we don't have a clear idea about the wild progenitor populations of mango, what I could do was look at this secondary bottleneck and try to understand whether mango suffered a loss of genetic diversity as it was moved out of India and into new regions of the world. So the two kind of primary goals of this project were to understand, is there a loss of diversity that occurred as mango was introduced into new regions? And are there regions of the world that have particularly high or unique genetic diversity? So I analyzed the same data set, this time focusing just on the mango cultivars that were included. So this represented a little over 100 individuals. And these were individuals representing each of these geographic regions. Everything here is color-coded according to the map. And there's multiple ways you can look at genetic diversity, all of these different measures. So I've summarized them here, but the main takeaway is that we don't see a lot of differences in levels of genetic diversity in these different regions. And we certainly don't see a clear loss of diversity coming when you're comparing India to other regions of the world. So it appears that mango has been able to maintain these high levels of diversity. However, if you look at diversity in a slightly different way and think about genetic structure, You can see on the right, this is a PCA. And you can tell here that the blue dots especially are showing up in a region of this plot that is, none of these other populations are showing up in. So this is telling us that these samples from Indochina, and then if you look closely, a few of the samples from Southeast Asia in green, and even a couple of the Mexican samples, likely those that were introduced directly from the Philippines, those come out as distinct. And so while these populations may not have a higher quantity of diversity, they certainly have unique genetic diversity, which is surprising because you wouldn't expect that if mango were originally from India. And so what this data shows is that this is really a tale of two mangoes. And this is something that if you talk to anyone who knows mangoes, they will say, of course, that makes a lot of sense because there are two different types of mangoes. There are Indian cultivars and there are Southeast Asian cultivars. And these can be differentiated based on their shape and size and coloration. If you go to the grocery store and you see the big green and red mangoes, those are Indian cultivars. And the little flatter yellow curvy ones are Southeast Asian. They differ in their flavor profiles as well, and also in a really unique characteristic of polyembryony. So Indian cultivars have a single seed with a single embryo, whereas Southeast Asian cultivars have a single seed with multiple embryos inside, one of which is sexually produced, and the others are clonal to the maternal plant. It's just a quirky little characteristic, but I think has some really interesting implications. Of course, the real question is, why do we see this differentiation? And this is a question I'm excited to dig into more, especially as travel begins to open up. I'm looking forward to getting back to this region to do some more thorough sampling of cultivars and then also wild species in the region. All right, so coming back to these goals of domestication genetics, I want to talk to you about specifically this third goal of using available diversity to improve our crops. I told you about how we know that a lot of times diversity is lost during this domestication process. And one of the main ways that we know we can combat this loss of diversity is by reintroducing diversity from wild progenitor species and crop wild relatives back into modern crops, just through traditional breeding techniques, hybridizing these individuals. And although this isn't a new idea, crop wild relatives remain an underutilized resource of variation that can provide adaptations to biotic and abiotic stressors. So as a postdoc working with Lauren Reisberg at the University of British Columbia, I had the opportunity to work on a project that puts some of these ideas into practice in the sunflower system. Sunflower is an oilseed of increasing global importance, and it's especially important in developing countries. This is work that involved a number of international collaborators and was funded by the Global Crop Diversity Trust. The first phase of this research involved the collection and survey of wild sunflower diversity. Many of these wild species are interfertile with the cultivated sunflower, Helianthus annuus. And these wild species are adapted to harsh conditions and can tolerate flooding, salt stress, and drought. So we were interested in trying to see if we could introduce some of these adaptations into cultivated sunflower. However, one of the main issues with using crop wild relatives in breeding is that these early crosses and producing the introduction process of this genetic material is pretty time consuming. And so oftentimes breeders aren't interested in doing it themselves. It doesn't tend to be worth it to them. And so while we have many collections of crop wild relative germplasm, it's not always being used in breeding programs. So the second stage of our project, Greg Bowdy, who was a PhD student in the lab, he created systematic crosses between 25 wild donors. He crossed them with a single line of cultivated sunflower, in total creating more than 400 pre-bred lines. And the goal of this was to create these pre-bred lines, stabilize them, so reduce sort of the variation within a line, and then hand these off to sunflower breeders around the world. So after creating these pre-bred lines, we did field trials and evaluation in collaboration with our colleagues in Uganda, Argentina, France, and India. And we tested for resistance to abiotic and biotic stressors, heat, drought, low nutrient conditions, along with important pathogens of sunflower. Well, we weren't just interested in finding individuals that could tolerate these conditions. We wanted to find individuals that could still thrive while combating these stressors and didn't suffer a loss in seed or oil yields. This project is still in the process of wrapping up, but we're excited that we've seen some really promising results. For example, in this graph, I've plotted two measures of productivity, oil yields, and kernel weight. Here, the kind of gray triangles represent the results for the 400 pre-bred lines, while the colored dots represent different check lines, basically controls. So some of these lines in blue were ones that we knew to be previously drought tolerant. Oh, and these were, this was an experiment for drought stress in particular. So the blue lines were known drought tolerant lines. We also have some known low nutrient lines. And in the yellow, we have the elite parental line that was used to create these pre-bred lines. And so what's interesting is that while a lot of these pre-bred lines don't necessarily perform well, or certainly not better than some of our cultivated lines, a few of them actually perform quite well and even better than the parental line. So this shows us that we were able to find some individuals that can still produce high yields under drought stress. We're also in the process of analyzing the genomic composition of each of these 400 lines to identify regions within the genome that have wild introgressions. So this is a figure of introgressions found on chromosome 1. And these are just the regions for which we had data. So there are large gaps between each of these regions where we don't necessarily have calls for whether or not there's an introgression there. The lines are colored by the family, so that is the wild donor plant. And the darker coloration indicates a region that showed evidence of introgression from these wild donors. So you can see that there's a lot of variation even within lines that came from the same parent in terms of which regions of the genome ended up remaining introgressed from these wild species. So we are currently working on finishing this up and then hoping to explore different regions of the genome that we know are associated with particular traits to see how those lines have responded in our field trials. So as I mentioned, the goal of this project was ultimately to provide sunflower breeders with new genetic diversity and data about these lines. And so therefore, all of these lines are publicly available through the USDA. And I'm really excited that all of the data that we've collected is also publicly available on the Germinate portal run by the James Hutton Institute. And we've actually already had a handful of sunflower breeders request this germplasm, and they've started to incorporate it into their breeding programs. So a spinoff of my work on sunflower in the Reesburg lab was work that I have been doing with Nolan Kane at the University of Colorado Boulder during a postdoctoral stint that I had there for the past year. And this is work focused on genomic structural variation within sunflower. So many of you probably know about structural variation within genomes, but in case you don't, the way I like to think about it is that if the genome is a book, we've been looking at single nucleotide polymorphisms, SNPs. So that's a single word that might have been changed or deleted or added into the text. Meanwhile, while we've been so hyper-focused on these word changes, there might be entire portions, pages of this book that have been ripped out, turned around, moved around within the book. And these are structural variations in the genome. So we're just beginning to kind of have the capacity to really dig into and identify these types of regions. So we don't have a great idea about how they respond to different evolutionary forces like selection, demographic changes, and introgression. And these are all things that happen during the process of domestication. So my work, the first question I'm looking at is how is structural variation impacted during this domestication process? And I'm particularly focusing on inversions, presence-absence variation, and copy number variation in elite, landrace, and wild sunflowers. And then I'm also interested in understanding to what extent structural variation contributes to important agricultural phenotypes, and especially in the mutational load that we're starting to realize many crop species carry with them. So this is ongoing work, but I'm really excited to see where it leads, and I think it'll have some really important implications for sunflower breeding, but also just our general understanding of the evolutionary dynamics of structural variants. So some of my other ongoing projects include a new project I'm involved with in collaboration with Colin Corey and Abby Meyer. And we're excited to be focusing on crop wild relative conservation in North American fruit and nut trees. This is a project that thus far is funded by Botanic Gardens Conservation International, BGCI, and the US Botanic Garden. This project really grew out of an analysis that Colin recently published in PNAS that looked at the conservation status of crop wild relative species in North America. So often we talk about crop wild relatives and we've thought about them in other parts of the world, but we really haven't spent as much time looking at the species that are in our own backyards. So I'm excited to have some of these local conservation projects to work on as well. So in this paper, The bottom right of the screen shows a heat map of crop wild relative taxa. And if you'll notice, where we are in Missouri actually has a pretty high number of these species. And then the top right, you can see, probably can't make it out super well, but the overall idea is just that they categorized these different wild species in terms of their conservation priority, with green being the least lowest priority and red being the urgent priority. And if you just look at the general trend, you can see most of the crop wild relatives fall either in this orange or red group, meaning that they're of urgent or high priority for conservation action, both in situ and ex situ. I was really honored to be asked to write a commentary on Colin's work. And of course, given my interest in woody and tree crops, I was interested to see how they compare to other species. So if you look at the crops that are grown around the world that have wild relatives in North America, about half of that list of crops are woody species. But if you look at the overall number of wild relatives found in North America, about 74% of them are woody species. And that number rises when you look at the percentage of urgent priority taxa. So essentially what this means is that woody species are overrepresented in terms of just their abundance in North America and in their priority status. And so our long-term goal for this project is to bring together communities from the Botanic Garden and germplasm gene banking communities to build efficient, multi-purpose meta collections and collaborative conservation targeting North American fruit and nut trees in particular. We've selected 10 genera, which you can see listed on the right, to begin focusing on. And this includes around 70 species. I'm really excited to see where this project leads in the coming years. And I'm hoping that over time, we'll be able to really integrate conservation with some phenotyping and genotyping of these wild relatives that can contribute to crop improvement of our native North American fruits and nuts. I'm also really thrilled to be part of a newly funded project, which is called the New Roots for Restoration Biology Integration Institute. This is a really large and diverse team that's led by Allison Miller, who's at SLU and the Danforth Center, who many of you probably know. And the goals of this institute are to advance the restoration of wild and agricultural landscapes, to improve prediction of below ground functional traits based on above ground phenotypes. And it will also help us accelerate the selection of robust perennial ecotypes for use in restored wild landscapes. This information will also be used for breeding perennial crops like intermediate wheatgrass and alfalfa, and will be used to help reimagine our agricultural ecosystems that mimic the diversity we see in the prairies around us. I encourage you, if you're at all interested in any of these thoughts about plant organismal systems, soil ecospheres, and plant population and community ecology, I encourage you to check us out on Twitter and see where our research takes us. We're also still working on building collaborations. So I guess in closing, I just want to I just want to come back to these 3 points of the focus of domestication genomics and how this work relates back to understanding the past evolution and domestication, how that's gotten us to the diversity we see today, what we can do to conserve that agrobiodiversity and then use that diversity in the future. So I'm really happy that I had a chance to talk to you about how my work relates to these 3 points. And with that, I'd like to extend a number of thank yous to folks throughout my career, from my time as a PhD student at Florida International University, working with Fairchild Tropical Botanic Garden, to my time at UBC and the University of Colorado, of course, all my collaborators and funding agencies. And then finally, really big thank you to my new employer, Missouri Botanical Garden. And I'm here for the foreseeable future, so I look forward to collaborating with those of you in St. Louis and beyond. Thank you.
All right, terrific talk, Dr. Warschefsky. So interesting. I'm sure that at home you were receiving a thunderous applause, and that is ringing throughout the streets and buildings of St. Louis if you were to just step outside. But fortunately, we have you here for a little bit longer on the Zoom call. And so I'm gonna go ahead and lead off and with some of the questions that we're starting to get here in the chat section. And this first question is from Dr. Susan Renner, and she is curious about some of the data that you showed earlier in the talk, when you sort of described how morphology doesn't really follow phylogeny, and how the sort of the morphological hypotheses don't match the phylogenetic ones. And so she's curious if that's perhaps a little bit too vague, and she was interested in what sort of morphological traits you were referring to in that case.
The morphological hypothesis was developed by taxonomists who actually described a number of the species within Mangifera. So this was, I wasn't using a particular morphological characteristic, but rather the entire classification of the genus based on the cumulative morphology of these individuals.
Thank you. That was a really interesting talk. So I have a question of my own, actually, like while we're waiting for the comments to roll in. So I'm interested in your Mangifera study. So among the species that you've sampled, is there any data on whether they're able to interbreed? And do you see any signature of introgression in those samples you collected?
Yeah, so that is actually, that's the chapter of my dissertation I left out because it's still somewhat preliminary data, I feel. But yes, so within the genus Mangifera, there is one confirmed hybrid species, and I think I have preliminary evidence of a second hybrid species, or hybrid, whether or not it's a species is debatable, within the group. And so that does contribute to this idea of there being a differentiation between Indian and Southeast Asian mangoes. One of the possibilities is that this genetic differentiation is the result of introgression from wild relatives when mango was introduced into Southeast Asia.
Thank you.
Really fascinating, thanks. So Dr. Natalie Mueller is wondering if the tolerance of the wild lines of sunflowers or their crosses to various stressors are sort of geographically patterned, and kind of where did the hardiest lines parents sort of come from?
Sure, yeah, that's a great question. I would definitely say there are some geographical patterns to that because, some of the species are geographically isolated For instance, the species that grow on the sand dunes in Colorado are fairly endemic to that region. But then within wild sunflower as well, there is some geographic structuring of tolerance. And it's debatable how much of that is I guess a response to the local conditions and adaptations, or sort of just, well, how much of that is actually plasticity versus the true adaptations, I guess.
Cool, thank you. So I actually have another question, kind of a more general question. So a lot of your studies are on woody crop species. Are there any, I guess, big patterns of differences between woody versus non-woody crops, like in terms of their domesticated histories or their genomic structure, or anything that you've noticed?
Yeah, so woody species are really unique because the entire process of domestication is quite different in these species. And that's largely due to some of their just biological traits. A lot of annual species and grasses can self-pollinate, and they reproduce every year. So you can inbreed these and create these true breeding lines, right? So the individual you collect the seed from, you plant that seed, it'll produce pretty much the same individual, or very morphologically similar individual. In woody species, that is not the case. If you were to take an apple seed and out of a Granny Smith apple and plant it, the tree you get would not look like a Granny Smith. It would not produce apples that look like Granny Smith apples. And that's because most fruit trees and many woody species are obligately outcrossing. And so the seed is highly heterozygous. And this is part of why these woody species maintain diversity during the domestication process. The way that we sort of get particular types of, or cultivars, like a Granny Smith in woody species, is that we're able to clonally propagate them using grafting. And so each Granny Smith apple is from a nearly genetically identical clone that has been moved around to different orchards.
Really interesting. This is so fascinating. You have a bunch of questions coming in now. Dr. Jonathan Losses is wondering if the domestication history of mangoes is typical of domesticated tropical fruits, and if you might be intending to continue working on the mangoes at all.
Yeah, I would say, I don't know for sure if I can answer if it's typical of tropical fruits, because honestly, there just aren't a lot of studies on domestication in tropical species. I would say it does seem to be to follow some of what we know from other tree species like apples and citrus. In general, there does seem to be a lot of hybridization that happens in these woody species that contribute to the cultivated species. And yes, I'm definitely looking forward to continuing to work on mango. I think there's a lot of really interesting avenues that remain out there. And it's obviously, it's a fun crop to work on.
Thank you. We have a question from John Birmingham. He asked, what constraints, if any, have you had in obtaining mangoes or sunflower samples from elsewhere in the world, given that the US is a non-signatory to the Convention of Biological Diversity?
Yeah, that's a good question. So some of my work, especially involving sunflower, that involved samples from the USDA. So those were already in the US. They dealt with importing that, and sunflower and most of the relatives that we were using for that project are all native to North America anyway. In terms of mango, the cultivated mango, most of those collections were from the Botanic Garden I was associated with in Miami. The wild species I was able to obtain through collaborations with gardens, primarily in Southeast Asia. So just kind of standard MOU paperwork to transfer leaf material. I did not introduce any living material into the US.
So I have one last question as we start to wind down here. And I was curious, so in the mangoes, you have these kind of two genetic lineages. And I guess I'm interested to know if there are sort of phenotypic differences within that Indian lineage that is sort of in so many different places now. And I guess, yeah, do you notice differences in the same kind of ways in terms of flavor profile and stuff that you do between the two kind of very distinct genetic lineages?
It's certainly not as pronounced as you do between the two genetic lineages. And there are perhaps some differences, but I would almost think that could be chalked up more to cultural preference. So a lot of the mangoes that you see in Australia, for example, look relatively the same because they have one mango that they like, and that's the one they tend to pick things that look like that. It's always really fun to talk to different people from different parts of the world, because every region seems to have their own favorite cultivar. And so I think that would probably be more of the contribution to what we're seeing with patterns of morphological diversity at that scale.
Awesome, thanks. Well, I think that's about all the time we have for today. So hopefully everybody at home is giving Dr. Warschefsky another big round of applause for a really excellent seminar. And I hope that you'll all join us for our seminar next week. Take care.
Thank you.